Howdy, this is Group 2 for Lab 3 Media. Why do we need some different media? Well, we need it for selective growth, differential identification, as well as functional testing. The media we use are blood agar, CNA blood agar, bile esculin agar, mannitol salt, eosin methylene blue, McConkey agar, salmonella shigella, motility, and thioglycolate medium. Our cultures are Alcaligenes fascialis, Clostridium perfringens, Enterococcus fascialis C, Escherichia coli, Klebsiella pneumoniae, Listeria monocytogenes, Mycobacterium smegmatis, Salmonella trifimurium, Shigella flexneri, Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus impiegens. Plates will be sectioned into 3 or 2 depending on how many inoculations are done. Motility tests will be done on a semi-solid with a stab to determine if the bacteria travels, and thioglycolate will be used to determine aerotolerance by grabbing bacteria and looping it in the liquid. Blood agar is enriched and differential, and it detects hemolysis, whether it be alpha, beta, or gamma. K. pneumoniae appears to have no clearing, which is gamma. SRS and E. fascialis both grew, and SRS does appear to have a clear zone. Our unknowns both grew, and it is hard to determine which ones had clear zones. CNA blood agar is selected for gram-positive and inhibits gram-negative. E. coli and S. pyogenes did not grow, however SRS did. For our unknown number 1 and 2, our unknown number 1 did grow, but our unknown number 2 did not. Bioesculin is selected for group D strep and enterococci tests, and tests esculin hydrolysis in bile. E. fascialis did grow and also had a black precipitate, and the other 2 did not. Our unknown 2 did have black precipitate and did grow. Our unknown number 1 did not grow. For mannitol salt agar, it is selected for staphylococci and differential for mannitol fermentation. SRS did grow and it appeared to be yellow, meaning it's a fermenter. The other 2 did not grow. And for our unknowns, they didn't grow either. For the McConkey agar, it is selected for gram-negative and differential for lactose fermentation. Our E. coli did grow and appears to be a pink color, which is a lactose fermentation. The S. trifimurium does appear to grow, but is not colored, meaning it is non-lactose fermentation. Unknown number 2 did grow and was pink, meaning it's a lactose fermenter. EMB is selected for gram-negative and differential for lactose fermentation, but apparently there was an error, not on us, but on the plates themselves. And E. coli should have been apparent with a green sheen, but there was K. pneumoniae and it was purple. EMB on the unknown number 1 and 2 did show growth, and unknown number 2 is purple. Salmonella and Shigella plates are selective for those 2 and detects H2S production. The E. coli and the S. trifimurium did grow, however the S. flexneri did not, and they appear pink as well as colorless. Our unknown number 1 and number 2, the unknown number 2 did grow, but the unknown number 1 did not.
For our results on the blood agar when testing for their aerobic properties, E. coli, A. facilis, and C. perfringens are pictured in these results below. The only one that did not experience any growth at all was the C. perfringens. However, the other 2, E. coli and A. facilis, did experience a linear type of growth. On the blood agar aerobic plates for the unknown number 1 and number 2 both once again experienced growth. However, one was quite linear and thin compared to the other type of growth that was pictured on the right side of the plate on the right photo, which was the unknown number 1. And this one had a thicker, linear growth. The blood agar anaerobic results are pictured below for E. coli, A. facilis, and C. perfringens. All of them did experience a linear, thin type of growth. However, you can see that there is a slight difference in the color hues, as two of them appear more white, and the one on the right, which is the C. perfringens, is actually more of a yellow hue. When we tested the unknown number 1 and number 2 in the anaerobic conditions, they both experienced growth, both linear. However, one was thin and one appeared to have a thick, cloudy-like appearance that did not seem as defined. And this was the unknown number 1 that was pictured on the right with the growth. When we look at our mediums, specifically the thioglycolate medium, the E. coli did experience some growth that appears cloudy at the bottom of the test tube. And so on the right side, we can see that this cloudiness was about an inch at the bottom of the test tube. A facilis in the same medium did experience a bit of growth. It is difficult to see in the picture, but midway through we could see particles that were visible once you were able to see it with the light. And so unfortunately it's not as visible here, but there are some particles that are visible in the middle of this test tube. The C. perfringens in this type of medium are as well also seemed to have a cloudy appearance at the bottom of the test tube, indicating that there was growth. And when we utilize the unknowns, starting with unknown number 1, we did see quite an interesting pattern of growth here in this test tube. It looks like an interesting type of cloudy appearance once again at the bottom, but this one does seem a bit different compared to the other two that had this similar type of growth. This cloudiness appears to be more defined and visible to the naked eye. Our unknown number 2, however, was not as noticeable, although the liquid medium does seem to have a more cloudy appearance. It was not as visible compared to the cloudiness that was in the previous unknown number 1. When we look at the motility mediums now with the E. coli, it was very interesting to see this type of growth. It was extremely linear and it was very visible. And so it was quite a successful type of growth that was seen, and we were able to capture it effectively through the photo. Once we go to the K. pneumonia in the motility medium, we once again were able to see the growth very well in the same linear fashion. Although the inoculation could have been more precise, as you can see that there are 2 visible lines of growth. However, we were still able to successfully grow them in this medium. When we look at the unknown number 1, it seems that some of the growth was visible on the side of the test tube, which could be a result of not successfully inoculating as precisely as possible, but we still see that there is a linear growth that can be seen specifically on the right photo. For the unknown number 2, this is the type of growth in this motility medium that is probably the least visible. We do see that there is a type of film at the top of the liquid where the motility medium starts. This does indicate growth, and with the different lighting, you can see that on the left side, you see the linear growth, and on the right side, you can see more precisely exactly how it grew. And this was the unknown number 2. In conclusion, when we look at the aero tolerance, the E. coli showed growth throughout, but it was heavier near the top, indicating that it was a facultative anaerobe. The A. facilis only grew at the surface, which showed that it was an obligate aerobe. The C. perfringens grew at the bottom, confirming that they were obligate anaerobes. And this truly reflects their habitats. For E. coli, we usually see them in our intestines, and for A. facilis, they are found in oxygen-rich environments. C. perfringens are usually in anaerobic tissues and soil. For the selective and differential media, we did notice that for the McConkey and EMB, it only allowed gram negatives to grow. Lactose fermenters produce a pink, red, or metallic colony, and the non-fermenters stayed colorless, which we couldn't tell with the naked eye. With the MSA, they were selected for gram positives, and the mannitol formation turned the medium yellow. And for the SS agar, this inhibited gram positives, so differentiated lactose fermenters, pink red colonies from non-fermenters, which were colorless. On the mannitol salt agar, the SRS grew with yellow medium, the mannitol fermenter, and then S. epidermidis growth was medium and it stayed pink or red because it's a non-fermenter. And S. pyogens, there's no growth due to the salt sensitivity. When we look at the thioglycolate medium versus the anaerobic chamber, we saw that with the thioglycolate results, the E. coli was grown throughout and it was heavier near the surface because it's facultative. The A. facilis was only at the surface because it's an aerobe, and the C. perfringens only grew at the bottom because it's an anaerobe. With the anaerobic chamber results, they matched the thioglycolate patterns, which once again confirms their oxygen requirements. The advantages and limitations of each include the fact that thioglycolate is simple, it's inexpensive, and it shows oxygen preference gradients, but cannot create a completely oxygen-free environment. However, with anaerobic chambers, they do provide the strict oxygen-free conditions, but they do require more commitment with time and equipment. Thank you so much for listening. We really appreciate this. Once again, this is Group 2, Diego Castillo, and myself, Rita Cantu Mahurin. Thank you so much.